Okay, hi everyone. My name is Anne, and just a fair warning, my project is very similar to Alex, so make sure you remember what he said. So my research was on determining if Micropterus dalamute or smallmouth bass are predators of endemic stream animals. So I chose to do this research project because I've been working with these endemic animals for 2 years now, and I'm continually interested in how we can detect species without ever seeing them. So as Alec had mentioned, there are 5 endemic fish species called oopu and 2 endemic shrimp species called opai. And so they're extremely biologically and culturally important to Hawaii. So native oopu are actually the most mentioned fish in Mo'olelo, and native opai were eaten as a delicacy and are also bioindicators of weather changes. So what's also special about these species are that they are amphigamous, which means that they're born in the ocean and then heavy rainfall or another weather event will wash them into, sorry, did I say they're born in the streams and then heavy rainfall washes them into the ocean, and then they spend the rest of their lives trying to migrate back upstream into their homes, into where they were born. So throughout this journey, they face a lot of different barriers, such as physical barriers and biological barriers. So from the ocean to the streams, they have to swim through our beautiful alawai. And from there, at the bottom left, you can see Palolo Stream, which is an extremely urbanized, full of cement stream. And so there's also some biological barriers, such as predators and competitors. So barriers include what actually interests me in this project is that Palolo Stream is much more urbanized than Manoa Stream. It's full of concrete and the temperatures in the water are often hot. Yet native oopu are more frequently found in manual surveys than in Manoa, and the smallmouth bass don't actually have a population in the Palolo Stream. And Manoa is a much cleaner habitat, but less oopu are found, and there is a large population of smallmouth bass. So from the Alawai, there are 2 streams that converge into one, Palolo and Manoa Stream, as you can see in that picture. So I was wondering if this presence-absence of smallmouth bass and oopu have a connection. And if so, are smallmouth bass eating these endemic species? So smallmouth bass are a very invasive predator that were introduced in the 80s due to sports fishing. They're super fun game fish to catch. And they're also very voracious carnivores. So contrary to their names, they will eat anything that can fit in their mouths, and that leads some people to believe that they can be predators. However, there's no real way to prove this. And so these are pictures I took after our dissection of the smallmouth bass stomach contents. And this is kind of what you see. You're going to need a lot of taxonomic expertise, and it's very hard to visually identify specific species. Like you can probably mostly get to the genuses of them, but it's extremely difficult to identify. So this is where DNA metabarcoding can fill in the blanks. So DNA metabarcoding can detect species molecularly through the remains of DNA in the stomach contents. So this reveals the presence of species that smallmouth bass directly consume without needing to ever see the prey in the stomach or identify it. So my goals of research were to first determine if DNA metabarcoding is a reliable method of determining smallmouth bass diet, and secondly, to uncover if smallmouth bass are eating endemic animals in our streams. So we sampled from 3 varying elevation from the same stream. So as I showed before, Manoa-Palolo confluence is where the 2 streams converge into one stream. And there are 3 species of native oopu that are frequently found here, the oopu okupa, oopu naniha, and nakea. And then I also sampled from Manoa Marketplace, which is at 160 feet, and Upper Manoa Stream, which is at 260 feet. And pay attention to these elevations because they do play a role later on. So these are the steps from collection to results, and steps include morphological identification, molecular lab work, and bioinformatics processing, which I'll break down in the next slides. So first, our smallmouth bass were collected by my mentor, Mr. Corey Yap. So he's a part of the Sangstream lab at UH and a part of Iolani. So shout out to him, and thank you for helping me collect my samples. So for sampling, we dissected 38 fish, 8 in the first trial, and then 30 in the trial 2 months after. And we took the stomach contents out of the smallmouth bass through dissection, in which Mr. Yap then looked over the stomachs to try to visually identify what he could. And then we then took like a 16th teaspoon of the stomach mush that's in there, and that's what we are extracting DNA from. So DNA extracting will extract all the DNA out of the stomach content sample and essentially take the concentrated DNA in the stomach contents through breaking down the proteins in the DNA strands. So this was done using the QIAGEN DNA extraction kit. And after the extraction, I used the PCR primers MyFish, MyDeca, and ArcCOI to PCR amplify the species from my samples. So PCR primers are group specific, so they amplify certain genes only. As you can see, like 12S for fish, COI for arthropods, which are like bugs and decapods, and 16S for decapods, which are like crustaceans. And so, for example, the MyFish PCR only amplifies fish DNA, and that filters out the rest of the DNA like pigs, humans, bugs, etc. So the PCR products are then loaded into a MinION sequencer onto a fungal flow cell, which is a fancy machine that is able to spit out all of the DNA sequences detected in the sample onto a computer. So the rest of the process includes bioinformatics processing and species identification. So I organized the sequences based on the species using some coding of like the R programming language. And we then blasted the DNA, which stands for Basic Local Alignment Search Tool. And this finds regions of local similarity between protein or nucleotide sequences based on the statistical significance of the matches. But to simplify those words, it's basically like using Shazam for DNA. So you throw in the sequences and it'll show you exactly what species are present in the DNA. So to answer my first question, can using DNA metabarcoding increase species detected in the smallmouth bass diet comparative to manual observation and dissection methods? And the answer is yes. So for our first trial, we sequenced 8 smallmouth bass stomach contents. And so as you can see, across these 8 individual fish, we're able to identify a lot more diet diversity than you are able to visually identify. And additionally, we found that 2 out of 8 of the smallmouth bass were directly consuming Oopu akupa, which is one of our native species. And this was taken at the Manoa Pololo confluence, which again was at that 40-foot low elevation site. And for my second question, are smallmouth bass eating endemic animals in Oahu? Again, yes. So this trial was taken 2 months after the first one in November, and we sampled 30 fish in total, 10 across each elevation site. And we used arthropod, fish, and decapod primers for this one. And we found that not only are they eating native Opokupa at the Manoa Pololo confluence, as well as the Manoa Marketplace stream, we also found that they're eating Opai Oaha'a, which is our endemic shrimp species as well in the 40-foot stream area. So as you can see, not only are they continuously eating these Oopuakupa despite the 2-month time difference, they are also continuously eating these Opai Oaha'a. And so there's also a difference in elevation. As you can see, they are not eating any endemic species at that 260 or 160-foot elevation, but they are mainly in the Manoa Pololo confluence area. So to reiterate what my findings have, DNA metabarcoding reveals more diet diversity and accuracy than morphological identification. And smallmouth bass are eating these endemic species, both Oopuakupa and Opai Oaha'a, and the Oopuakupa across both trials. And then again, there's predation correlation with elevation. So I learned a lot throughout this year as a whole. One of the main things I learned about was environmental ethics. So I had to have some really hard conversations about what does environmental ethics mean and how research can have an impact, like a good impact. And so obviously from my research, you can see that these smallmouth bass are really bad, right? They're eating all of our endemic species. And if these endemic species are gone, they're never going to be found ever again. They're only here. But also, these smallmouth bass have an extreme key role in the ecosystem, right? They're extremely carnivorous predators, and they take out a lot of our invasive species as well. So we don't really know what's going to happen if we start removal efforts, and like who knows, maybe another invasive competitor will be able to rise up and make it even worse for our endemic species. So having going forward with the removal efforts, we have to be really conscious about the effects on the stream health index. So I hope to do that later on. And another piece of advice for future researchers is to take pictures of everything, seriously everything, because you will be using them for science fairs, symposiums, conferences, and you would really like to have those at hand and not just have to Google them. And the most important thing, I think, is to try to understand your project in depth and not just do it. So everyone talks about choosing a project that you're passionate about, but I want to emphasize that this is meant to be a learning experience. So a lot of us may get caught up trying to finish collecting all of our data and trying to get good results, but we don't sit down to fully understand what it is that we're doing. And we have this unique chance to be this one student high school researcher who knows the most in the world about our research project. So I would urge future researchers to focus on the process of learning and fully understanding the limitations and implications of your project to truly create impact. And so lastly, I want to thank my mentors and my research class for their endless support. So although Dr. Chan can't make it today, I'll just talk to the camera. Thank you for giving me the best advice and for talking to me for hours about possible implications and limitations of my research. I cannot thank you for all the support you've provided me, and you've become such a role model to me and many others, and have definitely inspired me to become a woman in STEM. Mr. Hill, thank you for being the most hardworking mentor out there. I honestly feel like you could have just come up here and presented my project because of how much you know my project inside and out. So thank you for being just as passionate in this project as I was and never hesitating to fully explain all the molecularness behind it all. And Mrs. Kobayashi, thank you so much for being a role model to me these past 2 years. You've taught me so many skills last year that I was able to utilize more independently thanks to all of those PCRs we did last year. And you have truly inspired me to push myself and become the best researcher I can be. And Mr. Yap and Ms. Andy, thank you so much for your insight into my project. Having those deep conversations around these endemic species is really what brought this richness in my project and has instilled so much passion towards conservation within me. So thank you for your devotion to this line of work, and I hope to continue working with you to preserve these amazing animals. Oh my gosh. Thank you to Stream Lab and Jamie Hiji, who have helped me with my research in my lab and furthering my stream knowledge. And a special shout out to Period 2 for getting through this year with me. I couldn't have undertaken this project without everyone I mentioned. So thank you all for being here and listening as well.